Biology 400 students, this is Mr. Gales. This is Screencast Session 2 of Scientific Method and Experimental Design. Now remember, from Screencast Session 1 we looked at the goal of science, the steps of scientific method, how to write a good hypothesis, and we learned about the independent and dependent variable. In Screencast Session 2 we're going to talk about how to design a quality experiment. We'll look at the relationship between sample size and validity. We'll talk about data collection and how the different kinds of data that we might use in our experiments. And we'll talk about conclusions and their relationships to theories. And then we'll finish up with the characteristics of science, what makes science unique as a way of knowing. All right, now I want to remind you as we get started here that you need to take your 2-column notes from the video, and that the main ideas will be underlined and bolded. Those go on the left-hand column, and in the right-hand column you're going to put down definitions, examples, maybe lists, anything that will help you to remember or to understand the main idea. Also, it's really important that if you've got questions that you write them down as well so that we can talk about them in class. All right, so we're going to move forward into our first slide here. And the main idea that we're going to begin with, or kind of the big idea that we'll start off talking about, is a controlled experiment. Now remember, in the first screencast we saw a student named Jesse helping his teacher test a hypothesis about plant growth. The one characteristic of a quality controlled experiment is that it really allows us to test one variable at a time, and that would be the independent variable. If you remember, the independent variable is the variable that we believe is the presumed cause. It's what we think is going to make something happen. And in the video that we watched, Jesse and his teacher came up with the idea that the independent variable would be the amount of water given to the plant, or rather, how often the plant was watered. Okay, so that's a controlled experiment, an experiment that tests one variable at a time. Now, controlled experiments include two groups, and these two groups are really important to help us isolate these variables. Now, so the next main idea we'll look at will be the experimental group. The experimental group is the group that's exposed to the independent variable, and let's just do a quick review here. Remember that the independent variable is what we believe is the cause, right? So the experimental group is what we would give that variable to. The effect, or the outcome, what we measure as the scientist, is the dependent variable. Experimental groups in Jesse's experiment was the group that received water, I think it was every day. The other group that has to be found in every controlled experiment would be the control group. Now this is really critical. Control group doesn't mean that you have your hands on it and you're controlling it. What it means is it's not exposed to the independent variable, but all other variables are controlled and held constant. Okay, so in the video that we watched, both plants in the experimental and control group had to be the same kind of plant. They had to be placed in a similar position, so they had equal amounts of sun. They would have had to have the same type and amount of soil. Okay. Now, one other thing that we're going to be learning about here are called extraneous variables. These are variables that are also referred to as controlled variables because they have to be held constant in both groups. The value of making sure that you hold your extraneous variables constant is that way you really can tell whether or not the independent variable in fact has some sort of effect on your scientific subject. All right, so what I'm going to show you here is the second part of the video in which Jesse is working with his teacher, and they're really going to talk about this experimental group and control group, and he's going to get into some detail about the controlled variables. So let's go ahead and watch that video.
Well, would you look at that?
Wow, yeah. The plant that got more water is bigger.
So what does that tell you, Jacob?
It tells me that the one thing we did change, which is our independent variable, in this case the water, did have an effect on the plants.
And how do you know it wasn't something else? Like how much sunlight each plant received that made one plant grow bigger than the other one?
That's easy. We kept everything else the same. The same amount of light, the same place, the same temperature even. By keeping all the other variables the same, I can pretty positively say that by watering one of the plants more, in this case, actually caused it to grow bigger.
I would have to agree with you there, Jacob. And all those variables that you kept the same to keep the experiment fair, what are those called? Do you remember?
Controlled variables. But wait, wasn't there one last thing you wanted to tell me about?
Yep, that's right, Jacob. And the other thing is called the dependent variable.
Wait, before you tell me the answer, Mr. Koenig, let me see if I can figure out what the dependent variable is by myself. So, I know what dependent means. It means to depend on something. I depend on my parents to give me a place to live.
So far, so good. Keep going.
So, in our experiment, the dependent variable must be something that depends on something else. Like in our case, how tall the plant got depended on how much water you gave it.
That's right, Jacob. An easier way to think about it is like this. In an experiment, the dependent variable is what you're trying to measure. In our experiment, we were trying to measure how big the plants got. We found out that how big a plant got depended on how much water you gave it. So the thing that we changed was how much water. So that's our independent variable. The thing that we measured was how big it got. So that's our dependent variable.
All right, I get it. When I do an experiment, the thing that I'm measuring is my dependent variable. The thing that I change and test is my independent variable. Everything else should stay the same, and those are my controls.
That's right, Jacob. So for your next experiment, all you have to remember is that you need one independent variable, and remember, that's the thing that you change, one dependent variable, and again, that's the thing that you're trying to measure, and finally, keep everything else the same. You should have a fair test.
Awesome. Thanks for your help, Mr. Koenig.
All right, so we saw in the video some great examples of experimental groups and control groups, and we learned a little bit about extraneous variables, which were referred to as controlled variables in the video. Next topic I want to talk to you a little bit about is sample size. Sample size, which would be our next main idea, that goes on the left-hand side, is sometimes symbolized or given the symbol n, right? So the number of subjects, n for the number of subjects involved in the experiment. You have to keep in mind, if we look at this picture that's over here on the right-hand side, you may have a very large population that you're hoping to be able to do something with, right? But it might not be practical for you to test all of the members of that population, so you want to take a sampling. In this picture, we have our magnifying glass, which is focusing on a small group of that overall population, right? So your sample is the subgroup of your population that you're actually doing the experiment with. Depending on what kind of experiment you're doing, sometimes it's not practical to have a very large sample size. However, there is a general rule that you should be aware of. Increasing your sample size, in other words, increasing n, which is sample size, does a couple of things for you. First of all, it decreases the impact of chance events and error. Let's say we're testing the effect of some sort of new medicine on a population in terms of preventing high cholesterol. If you have very few test subjects involved in your experiment, there is a chance that there's going to be error involved, or that there will be chance events. Maybe somebody is going to get sick that's not related to their cholesterol. By increasing the sample size, you decrease the impact of chance events and error. Now, the other thing that results from that is you increase the reliability of your results, which we were going to refer to here as validity. And that's going to be our next main idea. So on the left-hand side, validity. Validity can be defined as the extent to which experimental results can be generalized to other settings or situations. I mean, this is kind of the idea of applied science. We want to be able to learn about how we can interact with the natural world. In order for us to be sure that our experimental results will work in the larger world, we need to have high validity. So to increase the validity, we're going to increase the sample size. So for instance, in Jesse's experiment, maybe we have 10 total plants instead of just 2 of them. We have 5 experimental plants and 5 control plants. We would hold all the variables constant except for the amount of water. And what that does is it makes sure that the growth of the plants is not going to be impacted by things other than the water. It's not just going to be a chance event that maybe one of the plants was sort of unhealthy to begin with. The picture here sort of represents for us the idea of generalizing results. If this is our sample of the larger population, if we design an experiment well, we should be able to take the results that we get from our sample, and we should be able to generalize it to a larger population and get the same results in a different setting. All right, let's talk a little bit about data. The main topic we're looking at is recording and analyzing data as part of the scientific method, but really there are 2 main ideas here, the different types of data that we may collect. Our first main idea that you see is called quantitative data, and if you look at the word here, quantitative has another word inside of it that you may recognize, quantity. Quantitative data is numerical data. It's, for instance, the measurements of the amount of water in milliliters that we may use. Or it could be a measurement of height in centimeters. All right? Quantitative data. This is the kind of data that we're most used to using. It's the kind of data that we use instruments to collect information about. The other type of data that we'll often use will be qualitative data. Qualitative data is data that is descriptive. It's not numerical in nature. This is the kind of data that you collect with your senses. So for instance, color. It could be a measure of how green the leaves on a plant are, right? Or it could be a measure of how healthy the plant looks based on the appearance of the leaves. Whether you're looking at quantitative data or qualitative data, a good way to organize your data is into some sort of table or chart, and that will help you to more effectively display your data so that we can analyze it. And by looking at this chart over here, we can see that plant A seems to have much better growth than plant B over time. In other words, we start off at essentially the same height, but plant A is growing to a higher height or a taller height, and it's doing it much more quickly. So we would assume here that plant A maybe is getting more fertilizer, maybe a little bit more water. Okay, when we have our data, what that allows us to do is develop conclusions about the experiments that we've run. This is really important. A conclusion will always refer back to the hypothesis. So when you write a conclusion, you want to make sure that you mention what your hypothesis was, and then you're going to use the words either supported or rejected. We're not going to use the words proven. In other words, you wouldn't say that the data proves my hypothesis to be correct. You would say the data in this experiment supports the hypothesis that the amount of water given is related to the height of the plant or how the plant grows. It's always important that you refer to your data, so you're going to want to bring some mention of the numerical values or the qualitative values that you've collected. Now, one mistake that students often make is they want to say the data proves my hypothesis. Now, the reason we can't really do that is in science we make generalizations based on what we can observe, and when we do an experiment, we're not actually observing every example of how plants grow, for instance, or every example of treatment with a particular medicine. So the data supports the hypothesis, but it doesn't really prove it. Now, conclusions are drawn based on experimental research. If the same conclusions are supported over a very long period of time with lots and lots of experimentation and observation, they become what we call theories. Scientific theories play a huge role in our understanding of the natural world. And what we're going to see here next is a really interesting video that sort of highlights the importance of theory and differentiates between theories in science and theories in everyday language. So let's take a look at that video, and then we're going to wrap up with the characteristics of science.
Isn't evolution just a theory?
It is a common misconception among non-scientists that evolution is just a theory, but I think that comes from them thinking that a theory is sort of a careless idea that really doesn't have a lot of evidence. It's probably wrong anyway.
In everyday language, a theory, it basically is a hunch or a guess, but in science it is not.
Theories are the heart of science. They pull together observations, laws, hypotheses, and inferences into coherent explanations for the great mysteries of the universe. In the 1800s, Charles Darwin observed the enormous diversity of life on Earth and set out to explain it. From the evidence he collected, Darwin developed his theory of evolution by natural selection, a theory that explains how new species originate.
If the globe has undergone such profound changes in its history, geologically, surely all living creatures must have changed with it to adapt to their new conditions. Well, otherwise they would have perished.
What theory does is it draws the ideas together, it draws observations together, and makes sense of them to allow you to make expectations. That's what theory is. And then we go and test those expectations.
People used to believe that the sun revolved around the Earth because that's how it looked. Over time, the steady accumulation of evidence supported a different theory, that the Earth and other planets revolve around the sun. And what about gravity? Have you ever said, "Oh, gravity, it's just a theory. I don't believe in it"?
A lot of people seem to think that you need to be able to see something directly in order to accept it as a good scientific idea. But in reality, all you're seeing when you see a book falling are the effects of gravity. You're not really seeing gravity itself. Gravity is something, in terms of gravitation theory, it's something that we have yet to fully explain.
Even so, we still rely on theories to make predictions that help us conduct our lives, to travel, to heal the sick, and to explore the universe.
Apollo 11, this is Houston, over.
Before anyone had ever traveled to the moon, NASA scientists applied gravitation theory to predict what it would be like to move in the lunar environment.
So-called kangaroo hop.
Predictions like these are called inferences. An inference is a conclusion you can draw from the available evidence.
If I place this cube on the table, and you have the 5 and the 1 and the 3 and the 6 and the 4, what would you say is on the bottom? Again, you have the 5 and the 1 and the 3 and the 6 and the 4. One can infer by looking at the data that's the observable data that number 2 would be at the bottom.
Charles Darwin inferred from the evidence available to him that all living creatures, including humans, descended from a common ancestor.
I think it's more like a tree. A tree of life. Each new species is a new shoot that springs from the parent tree. These new shoots branch out and develop in their turn, and so on. Some branches die out, others keep developing. The trunk, the ancient common ancestor.
Today, scientists continue to find evidence that supports Darwin's common ancestor inference in the fossil record and in genes. With new discoveries, details of scientific theories are refined, and scientists are comfortable with the notion that theories are constantly challenged and amended. That's the nature of science. If apples suddenly start rising, you can be sure that some aspect of gravitation theory will change. But science will not reject the very idea that there is a force called gravity. New discoveries may also refine elements of the theory of evolution by natural selection. But the notion that the Earth and its inhabitants have evolved from a common ancestor will not change.
I don't think any person with even a small amount of scientific background would reject cell theory or atomic theory or germ theory, that pathogens cause disease, just because we think of those ideas as theories.
The theory of evolution is no different. Like other scientific theories, evolution also helps us make sense of the natural world in which we live.
All right, so in the video, I think it made it very clear that a scientific theory is more than just a hunch. It's something that is a time-tested explanation for how the natural world works. And that really reflects the characteristics of science that we're going to talk about right here. So I'm going to go through and briefly touch on each of these and then give you a couple of examples. Characteristics of science. Number one, science and the findings of scientific research have to be based on the assumption that the natural world can in fact be investigated and explained in terms that we can understand. In other words, we have to be able to, if we're going to use science, we have to be able to tangibly explore and be able to explain in ways that people would be able to grasp. Science has to be based on the results of controlled experimentation and observation. So when we develop theories, they have to be based on that rigorous scientific method. The results of our scientific investigations have to be repeatable and verifiable. That means that if somebody else does the experiment in the same way that you did it, they should get the same or very similar results. That's what it means to be verifiable. Also, science must be refutable. If a hypothesis is not supported by evidence, it must be rejected or reworded. And we would start over with the scientific method. Now, some examples of how those first four characteristics come into play here, right? We talked about in order for a valid scientific theory, or for a scientific theory to be valid, there has to be some way that an observation or experiment could prove it to be false. So I'm going to give you an example here. Einstein's theory of relativity makes predictions about the results of experiments. These experiments could have produced results that contradicted Einstein, right? So we say that that experiment, or that theory rather, is falsifiable. We could in fact, if we found that there were experiments that did not correspond to the theory, that we would reject it out of hand. Now, I'm going to talk a little bit here about the idea that it has to be investigated. Characteristics of science is that the natural world has to be investigated and explained in terms that we can understand. Let's say somebody comes up with a theory that the moon is populated by little green men who can read our minds, and they're going to hide anytime anyone on Earth looks for them. They're going to flee into a deep space or hide anytime a spacecraft comes near. Right? I mean, that's an explanation for what we observe in nature. Right? We've never observed little green people living out in space. But the reason that this is not scientific is, I mean, it's not falsifiable. There's no way for us to actually see these little green men. These green men are designed so that no one could ever see them. So that kind of gets to the idea of what a scientific theory can and cannot do. And then I'm going to wrap up with the last characteristic of science, which is, I think, really important for us to understand as we go through this year. Science is not a faith-based way to understand the world. One doesn't have to believe in science, right? It's not a matter of believing in the germ theory, or believing in the cell theory, or believing in the law of gravity, or believing in the theory of evolution. Rather, one comes to new understandings by first-hand experience. When you do experiments, when you make observations, when you draw inferences from available data, you can see how all the pieces fit together to make an explanation. And those explanations, again, have to be based on these previous four characteristics, that we can in fact investigate and understand the world around us, that it's based on evidence, controlled experimentation, observation, that we should be able to repeat what we've discovered, and that we should be able to refute it if contradictory data is ever found. Okay, I think it's important to keep in mind that, right, there's nothing wrong with religious belief. For instance, like creationism. You watch the video with Bill Nye where he describes creationism as a way of thinking. But keep in mind that things like creationism are not considered scientific theories because they do not meet the criteria of what science is, right? They do not meet the characteristics of science. Creationism, for instance, tells us what happened, but it doesn't explain how, when, or why, or through what mechanisms. It cannot be proven false, right? Because you cannot disprove the existence of a divine creator. And the last reason why it's not considered a scientific theory is the nature of creationism means that the primary tenet, which is divine creation, can never be changed, much for the same reason you can't disprove it. So it's very important for you to understand the distinction between science as a way of knowing, which is based on empirical observation and evidence, and other ways of knowing. For instance, understanding and having faith. Okay, so that is our look at science. We'll be doing some investigating in class. We'll kind of practice with a setup of an experiment and draw some conclusions. And as we move forward through the year, we're going to apply what we've learned in these first two screencasts. So until we see you next time in class, signing off, this is Mr. Gales. We'll talk to you later.